Sathyabama Institute of Science and Technology.
Good morning, everybody. This is Jesia Persis Preethi, Assistant Professor from the Department of Biotechnology, Sathyabama Institute of Science and Technology. If we have to speak about rDNA technology, we need to mention about the importance of cDNA libraries. So, cDNA libraries have a very predominant role to be played if you have to synthesize genetically modified crops and if you have to synthesize cloning of the organisms. For all this, cDNA libraries have an important role to be played. So, what are cDNA libraries? cDNA libraries are the DNA collection of DNA fragments that have been synthesized from mRNAs with desired proteins. So, with the rise of diabetes, we have rising requirements of the insulin, and for this, in this context, it is very important that if we can identify the gene responsible for synthesizing this insulin protein, we can very well make multiple copies of gene. Therefore, by using this and cloning it into the desired organism, we can increase the production of insulin. So, for this purpose, we have this cDNA libraries, and cDNA libraries have a very predominant role to be played in rDNA technology cloning as well as genetically modified crops or modified organisms. So, cDNA libraries, in order to make DNA libraries, we can either use genomic DNA or cDNA, which is synthesized from the mRNA. And the process of creating this complementary DNA from RNA, it uses the process of reverse transcription and the enzyme reverse transcriptase. So, the mechanism, if we see, mRNA strand, the desired mRNA strand is used as a template, and the poly A tail, which is present at the 3' end, becomes the starter for the replication, in which the oligo DT primer is added, which forms like a primer to kickstart the process of polymerization. And for this polymerization, for the synthesis of DNA from the mRNA template, we need an enzyme called reverse transcriptase. So, in the presence of this reverse transcriptase, a complete newly synthesized DNA is synthesized, and this DNA, which is synthesized from the mRNA template, is known as cDNA. And cDNA, once we synthesize the cDNA, we need to get rid of the mRNA, and after removing the mRNA, we remove it with the help of RNase H, and after removing this mRNA, we make sure we add DNA polymerase, which synthesize the complementary DNA to the first synthesized strand, and therefore we can synthesize the double DNA strands. So, DNA polymerase is a predominant enzyme which helps in synthesizing the newly strand, that is second strand. So, this is how the DNA strands are synthesized. So, here the mRNA is used as a template, and by reverse transcription we synthesize the new strand, and from the first strand which is synthesized, the first strand of DNA, the second strand is produced. So, by this process we can synthesize multiple strands as we desire, and this is nothing but the whole process and the mechanism of cDNA library synthesis. So, in order to synthesize desired DNA, we can use the PCR to multiply the strands. cDNA synthesis is a enzyme mediated process which uses primarily two important enzymes, which is a reverse transcriptase enzyme and DNA polymerase. So, reverse transcriptase is very crucial in order to synthesize the DNA from the mRNA template, whereas DNA polymerase is crucial for synthesizing the new DNA strand from the first synthesized strand. And here we also need an enzyme called RNase, which helps in digesting the mRNA, which is used as a template. So, what are the advantages of cDNA libraries? cDNA libraries are used for gene expression analysis, and it is very invaluable tool for PCR and genetically modified organisms, and this is how we can use cDNA libraries for our benefit. So, thank you.